Hello, my name is Greg Macleod. My laboratory studies calcium regulation in nerve terminals and its impact on neurotransmission. We do this in Drosophila melanogaster, the fruit fly, an attractive model for neuroscientists because of its genetic malleability and simple, accessible nervous system. In this nervous system, genetically encoded fluorescent indicators are available to detect calcium changes in nerve terminals. However, these indicators have limited sensitivity to calcium and often show a nonlinear response. Synthetic indicators are better suited for measuring the rapid calcium changes associated with nerve activity. We will demonstrate a technique for loading dextrin-conjugated synthetic calcium indicators into live nerve terminals in Drosophila larvae. Dextrin conjugation helps prevent calcium indicators from being sequestered into organelles such as mitochondria. The loading technique can be applied equally to larvae, embryos, and adults. We will dissect a larva in a small Silgard bath on a glass slide. The blue dental wax will support a glass loading pipette. Take a wandering third instar larva from the side of an uncrowded vial. Place the larva in several drops of chilled Schneider solution and pin it through the head and tail with the dorsal midline upmost. Use a sharp pair of microscissors to nick a small incision between the trachea, large enough to allow the bottom blade of the scissors to penetrate. Cut through the cuticle along the dorsal